Hi, everyone, and thank you for joining me. I'm Dr. Michael Lindinger. I'm president of the Nutraceutical Alliance, which is a contract research organization that does research for companies developing ingredients and developing products. And some time ago, I did a little bit of research with Lallemand Animal Nutrition, and I continue to do some work with them from time to time. So this is my company here. So welcome. So what we're going to talk about today are nutritive antioxidants. We're going to talk about what they are. We're going to talk about what they do. And what they do specifically in the context of oxidative stress. So we'll talk about what oxidative stress is, why does oxidative stress occurs, and what happens when we have too much oxidative stress. And in general, when we have too much oxidative stress, we end up with protein degradation, membrane degradation of cells, and ultimately that's what results in inflammation and pain. And when we have this ongoing over the long term, over what's called chronic periods, this can lead to disease. And I think already you're starting to get the sense, well, maybe nutritive antioxidants can act on this to help control or slow progression of degenerative processes and disease. And indeed, that's what we're going to try to do. We're going to see how we can control oxidative stress through nutrition. And we're going to do this very simply and very briefly. It's not intended to be in-depth, but we're going to discuss and demonstrate some general principles, and then we'll have some conclusions at the end. So the focus of this presentation is going to be on our companion antioxidant really is a product that can be added to the food. And we're talking really about the normal diet, or we can talk about a supplement. But we want to give this food an additional function that supports antioxidant status in the body. So when we talk about a nutritive antioxidant, we're actually trying to put it into the food to make it more functional. So there are companies now that are putting nutritive antioxidants into pet foods to make to give this additional function to the food. And the function that we're interested in is functions that can promote health and well-being or disease prevention. So nutritive antioxidants, what we're trying to do is reduce the oxidative damage in cells and tissues. Well, what's oxidative damage? So we just start first of all with nutritive antioxidants. We can start with a lot of ones that we already know about. So we might not think of them that way, but they do have antioxidant activity. Things like vitamins A, C, and E. Some of the trace minerals: selenium, copper, zinc, and manganese. Creatine and N-acetylcysteine. These are two compounds that have been used a fair bit in the sports medicine arena, humans versus dogs, and they do help muscles. But creatine is often not used as an antioxidant, but it does have antioxidant activity. And then we have a large number of compounds, and these are categories of compounds. And within each of these categories, there can be dozens or even hundreds of other specific molecules. The curcuminoids, the anthocyanins, those are found in berries. Curcumin is in turmeric, for example. The catechins and cryptoxanthines that are found in your teas and your coffees. Flavonoids, lycopenes, and polyphenols that are found in many of our fruits and vegetables. So all of these are very beneficial. Together, these are considered to be secondary antioxidants. So they're present in good foods and good whole foods, and they're present in supplements. You can buy them as standalones in supplements or blended supplements. So these are secondary. One of the things about secondary antioxidants is that each molecule of a secondary antioxidant is really only able to neutralize one ROS, one reactive oxygen species. So that's an important principle to keep in mind. So that means for each reactive oxygen species that our cells produce, we, in essence, need one of these to counteract it. We have these antioxidants up here that are primary antioxidants, and SODs are superoxide dismutases. We also have catalase, and we have glutathione peroxidase. So these are the three primary antioxidant systems. They are all enzymes, which means they catalyze biochemical reactions in the body. And these enzymes, in fact, are all made within our bodies. They're part of our innate natural defense system. They're part of the immune system. And in general, if we look at the effectiveness of secondary and primary antioxidants, we have increasing effectiveness as we go up this way. So the nice thing about enzymes is that they are catalysts. So that means they're not restricted to just one reaction the way these secondaries are. They're actually able to neutralize thousands or tens of thousands or even more of reactive oxygen species by just recycling through the reactions that we'll look at in a moment. Well, why do we get oxidative stress? Well, it's because we live in an environment where 21% of the air we breathe is comprised of oxygen. And oxygen is reactive. It's one of the things that we require for our bodies to live. All of our cells require oxygen for their metabolism. All of our cells have mitochondria, and it's the mitochondrial respiration, the oxidation of carbon in the respiratory chain of the mitochondria that produces reactive oxygen species as a side effect or a side reaction to the production of ATP, the high energy molecule that our cells need. The reactive oxygen species can normally be handled by endogenous antioxidant systems, and those are usually the primary antioxidants, those three enzymes that we were in. Normally, this is easily handled by the body. But we can have situations where the stress is excess. And by that I mean the stress exceeds the ability of the antioxidant systems, which means that the rate of production of the ROS, or the duration of production of the ROS, exceeds the ability of the antioxidant systems to get rid of the ROS. So that's either through a very high rate, or i.e. a very high intensity type of exercise, a very high intensity stress, a lot of trauma or a lot of exercise, or very long term, either one of those. The end result of reactive oxygen species acting on our cells is pain, damage. And when we have damage, we always have inflammation, and pain is part of the inflammation. It lets us know that we're healing. So in a way, that's not a bad thing. And in a way, we also need to have some damage and some repair going on all the time. That's part of the adaptation process, as part of the exercise training process. So when we're looking at systems to mitigate oxidative stress in the body, we have to find a balance between turning it right off and keeping it on so that we have a normal but not excessive level. Well, what are the reactive oxygen species? Let's take a little look at these. We have superoxide radical. And superoxide radical is two oxygens that have come together. And they have this negative charge. And this negative charge is what makes it a radical. A radical is when the charge balance of the molecule is not what it normally is. And this is unstable and very reactive. It wants to react with things. And because it is very reactive, and when it reacts, it causes oxidative stress because of the oxygen. It breaks it down, breaks things down, which is not good. Now, if we take one of those oxygens and we stick a hydrogen or a proton on it, we have a hydroxyl radical. And normally, this will have a negative charge. But you see here, the hydroxyl radical does not have a negative charge. And that's what makes it a radical. It makes it very reactive. And again, the oxygen reacts with other molecules and will oxidize and damage them. And if we have two of these that come together, we have hydrogen peroxide. We're familiar with this because you can buy it at the pharmacy. You can use it to disinfect and so on. But it's probably something that we really shouldn't use unless it's an extreme situation. Again, it oxidizes tissues, and it can be very damaging. So the biochemical reactions like this, and this is oversimplified. These actually go in both directions. These all the arrows here actually go in both directions, but we're trying to force the reactions in this direction because we have a lot of oxygen. We have oxygen coming into our cells because we're breathing air in. And the mitochondria, through production of ATP, through a respiratory chain, are producing superoxide. And when the system is operating normally, we have the superoxide dismutase acting on the superoxide to produce hydrogen peroxide. And then the hydrogen peroxide is in turn acted on by catalase and glutathione peroxidase and other peroxidases, but those are the main two. And it converts them into water, so it neutralizes them, and this is completely beneficial. It's good. So this is really what we're talking about the endogenous antioxidant systems and activating these. Now, because they're enzymes, we can increase activity both through increasing the rate at which they operate, each one, they can be modulated, and by increasing the numbers of each of those proteins, and both of those can happen. So when we're talking about stress, we're looking at some kind of a cascade like this happening. We have cells within our bodies that through these enzyme systems and respiratory chain, the NADPG oxidase system and the respiratory chain in the mitochondria. So I put skeletal muscle here with these immune cells, the macrophages, the monocytes, the neutrophils, because with stress, skeletal muscle is 40% of our latent body mass, so it's major. And these immune cells can produce reactive oxygen species at very, very high rates when they're stimulated. So it's these cells that really contribute to increases in ROS in the body when we have high rates of stress. ROS ends up activating inflammasomes. These are molecules in our bodies, such as NLRP3, and these in turn, this is a cascade system, activate stress-activated kinases, and these stress-activated kinases act on receptors on cell membranes in many cells through these signaling pathways, the ERK, the JNK, and the P38 signaling pathways, and they stimulate transcription factors. They turn on genes in those cells that produce these types of proteins, these types of molecules, the NF-kappa beta and activator protein 1. And these in turn stimulate the production of the pro-inflammatory cytokines and enzymes. So you may have heard of these pro-inflammatory cytokines, the interleukins, interleukin 1 beta, interleukin 6, interleukin factor alpha, cyclooxygenase inhibitor, or cyclooxygenase 2, and nitrogen oxide synthase, which produces nitrogen oxide. So these are important signaling molecules. Nitric oxide itself is an important marker molecule and a signaling molecule. And when we see increases in nitric oxide in blood or in synovial fluid, we know that there is an increase in oxidative stress in those tissues because this has happened. And these can further activate microglia, can activate macrophages. So what you see here is this cycle. And this cycle can continue and continue, and it become what's called a very vicious cycle that becomes relatively uncontrolled, and we end up in a state of disease where we have immunosuppression. So we want to not have this go out of control. We want to be able to regulate that, and that's ultimately what we're trying to do with nutritive antioxidants and through eating good foods. So if we look at the oxidative stress inflammation relationship, we're looking at something like this. This is a cartoon of some tissue. Here we have a blood vessel, and here we have a disruption in the cell. And it could be from reactive oxygen species that have acted on the membrane and damaged the membrane, and this allows pathogens to leak into the cells. We have release of molecules that come in, they infiltrate, and we get this inflammation. So that's normal. That's going to help the damage to heal. But things can get a little bit out of control if the reactive oxygen species are too high. There's too many signals. We're locked into that cascade, and we have excessive production and deficient antioxidant defenses, and we can get some severe antioxidant damage. So we don't want that, because we know that when this happens, for example, with muscle, we have very sore muscles that are sore for a long time. Or we have very sore joints and we end up with joint disease. So those are the things that we're trying to mitigate. So let's talk about normal systems, and exercise is normal. So I've done a lot of work with exercise, and it's a great way of stressing our bodies, a great way of pets and our companion animals stressing their bodies. But exercise, because there's an increase in metabolic activity of the cells, is an oxidative stressor. And we know this from many. So what we know is that exercise results in oxidative stress. And when the oxidative stress is high, we have leakage of the muscle membrane, the skeletal muscle membranes. And we know this because we can measure the concentrations of proteins that are normally only found in muscles. And we can measure them at increased concentrations when they're present in blood, things like creatine kinase. And we'll talk about a little bit more about that later on. So we know that with very high intensity exercise, and with long duration exercise, we can get muscle membrane damage because we have evidence of that in the blood. Well, let's have a look at what happens with muscle during exercise. We have the muscle contraction relaxation cycle that we're all familiar with. We need that for locomotion. It's what makes our limbs move. And this contraction and relaxation cycle requires energy from metabolic fuels. Those are the macronutrients that we talked about earlier, the carbs, the fats particularly, but also the proteins and the amino acids to some extent. So the short carbon molecules that are derived mostly from fatty acids and carbohydrates, these are transported into the mitochondria, and the carbon is then used in respiratory chain. And we have the production of ATP. Okay, so the production of ATP, of course, the demand for ATP becomes very, very high when the intensity of exercise is very high, and the demand for ATP is very long-term if we have long-term exercise. So you can see the relationship between the requirement for oxidation going on in the mitochondria, the production of ATP, and ultimately the side reaction that generates reactive oxygen species. Muscles and other cells are always producing reactive oxygen species. It's always going to happen. We can't turn it off. We actually don't want to turn it off because it helps our cells and helps our bodies adapt over time. And this is some of the things that we're very interested in doing. So normally the ROS can be very easily metabolized by the free radical scavenging systems. Those are the superoxide dismutase, the catalase, the glutathione peroxidase. When metabolic rate is high, and ROS production increases, this can exceed the rate at which the ROS can be degraded naturally. And the ROS are highly reactive and they oxidize many structures within the cells. And we have muscle damage, leakage of muscle enzymes into the blood, loss of cellular integrity, calcium activated apoptosis mechanisms, programmed cell death happens. And ultimately we need to have a large inflammatory response to clear away all this damaged cell debris and to have a resynthesis process, a regeneration of the muscle or of the damaged tissue occurring. So the things that we can measure in blood that indicates muscle damage are things like creatine kinase, aspartate aminotransferase, alkaline phosphatase, gamma-glutamyl transferase. These are typically associated with the liver damage when you're looking at a clinical test result. But when it's associated with long-term or long-duration exercise, these are almost always coming from muscle. There may be some coming from liver, but they're also coming from muscle. So typically these will increase in concert, and that's due to muscle damage. The greater the concentration, the greater amount of muscle that's been damaged, which is a reflection of a greater metabolic activity and a greater oxidative stress. The concentrations can continue to increase for up to 96 hours after exercise, which means that there's this ongoing degradation of damaged tissue, of damaged muscle. That's 4 days after exercise. That's with very severe high intensity type of exercise. So that's the kind of work that we want to avoid, because when we have this much damage persisting for that long, there's going to be a lot of damage, there's a lot of pain, and we've exceeded the systems excessively. This can be very, very damaging, take a lot of time to repair from, take weeks. So the reason it's going on for 4 days is that cells just continue to die and die and die. But it happens. So when it happens, or even not even to that extent, we're familiar that there's muscle weakness, there's muscle soreness, and that's not what we want. A little bit is fine. It induces cell regeneration, bigger muscles, muscle fiber type transformation, all the beneficial changes associated with training. So what we want is what's going down here on the right side of your slide: oxidative metabolism, increase in oxidative stress, normal oxidative stress, mild inflammation, beneficial tissue repair, stronger tissues, stronger joints, stronger muscles, everything. When we have excessive oxidative stress, we start going down this road, and we have excessive production of oxygen-free radicals, damage of the membranes, we have severe inflammation, muscle damage, and soreness, muscle weakness. And we want to use antioxidants to try to slow down right here. We don't want to turn it off, because we don't want to turn this off, but we want to slow it down so that we're operating more on this side of the slide and not over here in the vicious cycle side of the slide. Well, let's look at some of the evidence from research, from actual research. And melanoidaldehyde is one of the compounds, one of the molecules that you get when you oxidize lipids in cell membrane. So it's a marker of cell membrane damage, and it's not specific to any type of cell, but it's a general indicator. Here at T0, that's our baseline before high intensity exercise, and T1 is a warm up, and T2 is right at the end of high intensity exercise. And you can see that there's a significant increase in the amount of MDA present in the blood, indicating a significant amount of membrane damage. And it's still elevated 18 hours later. Again, this indicates that the membrane damage, that there's some ongoing damage. The stress has not been fully mitigated. So the inflammation that's occurred as a result of this is continuing, and we're still getting some breakdown of cells. If we look at glutathione peroxidase, or sorry, this is just glutathione, we see that there's a big increase in glutathione, which is another oxidative molecule. But we can see that that an upregulation of antioxidant systems has brought this back down to normal, which is good. But if we look at some markers of tissue damage, so I talked about creatine kinase, aspartate aminotransferase. Well, LDH, or lactate dehydrogenase, is another enzyme that's found in tissues. LDH isoform 3 is found in lungs, kidneys, and spleen, and LDH5 is specific to skeletal muscle. And we see that LDH3 is increased 3 to 4-fold in blood right at the end of exercise and 18 hours later, and essentially the same is true for LDH5 in muscle. And this indicates that there's most likely some oxidative damage that's occurred to the membranes, making them leaky, and it's most likely the lungs and the spleen, because this is horses. The kidneys are relatively inactive during the period of exercise. But in muscle, we see this doubling or tripling. In some horses, it goes very high of LDH, indicating muscle damage. DOMS, or delayed onset muscle soreness, is a direct reflection of the heavy exercise, the ROS production, and the tissue damage that happens when we have excess oxidative stress. So this is okay to some extent. We have regeneration, healing, and improvements, training effects. But DOMS really shouldn't be lasting for more than about 12 to 24 hours. Longer than that means we're not exercising the right way. So with exercise, we have increased metabolism, increased oxygen demand and consumption, therefore increased oxygen free radical production, therefore increased oxidative stress, and therefore we can have damage through reactive oxygen species on cells. And this results in increases in our markers of muscle membrane and other membrane damage, the MDA and also the T-bars, muscle damage, soreness, weakness, activation of repair mechanisms. So let's briefly look at joints. And in joints, the situation isn't that different. And joints is very important in dogs and cats as well as horses, because we do have high performance dogs. But one of the things that really impacts joints is overweight and obesity, which is increasingly becoming a problem in our companion animals. We're killing them with kindness, some people say, because we're giving them too much of the wrong kinds of foods. But when this happens, there's a very strong correlation between the chronic inflammatory whole body responses that we get with overweight, obesity, and our more rapid progression of joint disease. So we want to mitigate that as well. So let's look at joints, and we want to keep them healthy. So again, what we want is this side of the slide going on. We have oxidative metabolism, and we have lots of cells that are involved in joints. We have the chondrocytes, those are the cells that are in the cartilage that lines the bone, right? And we also have the osteocytes, the bone cells that are underneath the chondrocytes, and all of these are contributing. We have synoviocytes that are present in the synovial fluid, the fluid within the joints and within the synovial membrane that surrounds the joint. And there are a number of different cells in the synovial fluid as well. But when we exercise, when there's increased compression on the joints, there's an increase in the activities of these cells, and that results in an increase in oxidative stress. And if it's normal, it's all good. With repeated trauma and repeated microtrauma, if it's excessive, we can get excessive inflammation, and we can get excessive oxidative stress. And these acting together come down through here, and we end up with tissue degradation, cartilage breakdown, and ultimately degenerative joint disease. So we don't want this. Once you're past, there's different stages for degenerative joint disease. There's 4 clinical stages, and essentially once you're past stage 1, there's no going back. So you can sort of stop at stage 1 and not have it go worse if you can improve things through modified activity, through nutrition and nutritive antioxidants. But you can't go back to a completely normal joint. So we want to avoid this if at all possible. So if you have a high performance dog or a high performance horse, and they're doing a lot of exercise, there's a lot of stress on the joints, we want to be providing those animals with nutritive antioxidants before there's an injury that happens, because when we know that if that happens, it's really hard to go back. So as I mentioned, we have this normal cycle of extension, compression, flexion, relaxation occurring in all the joints. There's a normal wear and repair cycle. And as you progress through training cycles, you don't go to this in 3 days. It takes months and months of training. You end up with stronger tissues, stronger joints, everything is good. You have regeneration of synovial fluid as part of the compression relaxation. Exchange of fluid through the synovial membrane with extracellular fluid in the body. You have regeneration of articular surfaces during recovery periods from these very stressful periods, and the joints continue to function properly. It's exactly what we want. But when it's excessive, we end up with inflammation, and inflammation in the joint is very damaging. You end up with pain and swelling. You can feel the heat on the joints in your dogs and your horses. You can actually feel it when you palpate it, when you touch it. And the inflammation disrupts the normal wear and repair cycle and induces destructive signals and enzymes, activates destructive enzymes. And we end up with a leaky synovial membrane, cartilage breakdown of the articular cartilage, and the development of bone spurs. So in this diagram here, you can see this osteophyte, this dimple. This is called a bone spur. These can be very, very painful. There's some erosion here on the inside, and you can see the erosion of the articular surface here. And this is almost down to the bone in regions here. So this is what we're talking about when we're talking about cartilage degradation. So when that happens, you end up with bone on bone, and you don't want that. So again, it's a very complex, even more complex because there's more tissues involved. You have your synovial membrane, you have the synovial fluid, and then you have your cartilage that's lining the bones within the joint. And all of these are communicating with each other. And they're communicating to maintain, ideally, a normal, healthy state. But when the stress is excessive, you end up with excessive production of nitric oxide. Remember the nitric oxide in that vicious cycle. And you have this pro-inflammatory cytokine prostaglandin E2, and it's produced at very high rates and increases in concentrations. And these two are two of the big players that end up resulting in breakdown of the cartilage in the joint and an increase in the leakiness of the synovial membranes. So we want to try to prevent that. And when this happens, when you have breakdown of the cartilage, we have this increase in the concentration of glycosaminoglycans or GAGs within synovial fluid. So again, we can measure that. So what we're trying to do is prevent this degenerative joint disease from happening, where we have this osteoarthritic joint, where here we have bone on bone. The cartilage is completely eroded away in places. We have a very thin synovial fluid between them, so there's no lubricant. And in this kind of severe condition, which some people and some animals have, it's very painful to move. And so we definitely won't want to progress to this, but we do see our animals and people with that situation. What we want to try to maintain is this, and we can achieve trying to do this through nutritive antioxidants. So acute stress as well as chronic stress both impose severe demands upon the stress systems. Our nervous systems are always stressed to some degree. So they're stress systems within our bodies, and it is an important contributor to unwellness and poor performance. And the more different types of stresses we have acting on our bodies at the same time, then they sort of build up. They have additive effects, and so we need to look at everything together is the best way. So how can we use nutraceuticals to prevent or minimize excess stress and strain, with our focus on nutraceuticals being those ones that can have an antioxidant effect? Nutritional deficiencies and inactivity, because they often go hand in hand, they generate a lot of health and performance concerns. So they're responsible for the overweight and obesity that we see, and that's often accompanied by cardiovascular problems and joint disease. There's a lot of strain on our systems, and in terms of performance and in terms of health, the animal isn't allowed to achieve its genetic potential. It stays, weak and suppressed. But we provide beneficial nutrients in our foods, functional foods, nutraceuticals. We can use these to address health and performance concerns. We can help the animals achieve their genetic potential and achieve high performance. And whether that high performance is just maintaining a very high level of health or an optimum level of health for the activity that it's performing, even if it's just your pasture horse, or if it's just your cat that lives in your living room. We want to achieve a high level of health. So nutritive antioxidants, what do? Well, what we're trying to do is mitigate what happens when these mitochondria in our cells are producing a lot of reactive oxygen species, where we have oxygen coming into the mitochondria. That's needed to produce the ATP. The side effect is producing these reactive oxygen species. But these, when it's excessive, has this effect, damaging effect on proteins and cell membranes. This is a cartoon of the cell membrane. and you have damage, pain, and inflammation. So what we want to do with the nutritive antioxidants is block here and block here. We want to inhibit. We don't want to completely turn it off, but we want to control it so that we can reduce the amount of damage, pain, and inflammation. So again, the secondary antioxidants can come in and they can deal with superoxide and with hydrogen peroxide and hydrogen hydroxyl radical. But the main ones that we're interested in are using these primary antioxidant systems because they can be used over and over and over again, and they just keep working away and reducing the amount of these that are being produced or that have been produced. So nutritional deficiencies, they can be addressed. We can address health and performance concerns. We can address and try to mitigate chronic inflammation and high oxidative stress. We want to prevent a suppressed immune function because when this happens, we have all kinds of problems: GI ulcers, gastrointestinal ulcers, lameness, respiratory problems, cardiovascular problems, kidney problems, liver problems, overweight, obese, and cognitive decline, a decrease in our mental functions. So nutritive antioxidants are aimed at every part of our health systems: the immune health, which interacts with all of these main systems and all the other ones that we don't even think about. Well, let's focus now a little bit on Melafeed and these enzyme systems. So superoxide dismutase, as I mentioned before, converts superoxide to the less toxic hydrogen peroxide, even though it is toxic, and catalase and peroxidase later breaks that down. There's three main types of superoxide dismutases in the body. Within the cells, there's one isoform, the SOD1. Within mitochondria itself, there's another isoform. And within the fluid outside of cells, there's yet another isoform. This tells you how important superoxide dismutase is. It tells you how important the endogenous antioxidant systems are, because we need to protect our body fluids at each of those levels. So if we can do things that will turn on these systems in our bodies, that can be very beneficial for us. These, when they are turned on, also activate the other antioxidant enzymes, the catalase and the glutathione peroxidase. So these are very important molecules. Using Melafeed, Carillon has done a very extensive series of studies over a period of roughly about 10 years. But in 2013 published this paper. And in this paper, they sort of lay out a putative mechanism for how this really unique product, Melafeed, works. The nice thing about this, or one of the interesting things about Melafeed, is that it does contain these enzymes, and it does contain them at very high concentrations compared to almost all other plant products. There's only one other plant product that I know of that has values that are as similarly high as this, but the idea is that this happens. The problem is with these types of enzymes, they're large molecules of proteins, and they have low bioavailability, which means that when you eat them, you don't get very much of it showing up in the blood. Well, that's because they are proteins, and so they are broken down by acid and by proteases in our stomach and in the small intestine. So this is true of dogs, cats, and horses and people. So it's broken down. So it's not these enzymes that are going into the body and exerting their beneficial effects. But one of the things we know from Carillon's work and the work of others is that when animals started with some studies with mice, and now we know that this happens in people as well, when they take this product, we get increases in the concentrations of these antioxidant enzymes in our bodies. So it's somehow causing this effect, even though it's not being absorbed. So they are ultimately degraded in the GI tract, and they're rapidly excreted by the kidneys. So to get around this, what Bionove and Lallemand have done is they encapsulate their melon product in a shell, in a microcrystalline shell. vegetable casing, and it protects the product as it goes through the stomach and the small intestine, so it reaches the large intestine intact, where it can the shell, the case dissolves, is eroded away from it, and there it starts to exert its beneficial effects on the animal. So again, these beneficial effects were first seen in mice, and they saw increases in SOD activity, and we see this now in horses as well. We'll talk more about that later. So the interesting thing is that these exogenous SODs, they're provided in foods. They have this additional influence that's independent, but somehow related to their antioxidant activity, because the antioxidant activity that they have can really only have their effects within the large intestine, which is going to be certainly beneficial, and all the indications are that it is very beneficial. Because there's the intestinal system, which we're not going to talk about, is a very highly metabolically active system, producing a lot of reactive oxygen species. But what it does, importantly, is it induces the endogenous, it stimulates the endogenous antioxidant defense systems, which is very, very cool, very important. So, and you seem to also have this intestinal activation of the immune system. And the innate immune system is an integral part of the intestinal system. They operate together. It's a unified system. And if you have intestinal damage, then you're almost always going to have some kind of immune suppression going on at the same time. You take care of the intestinal system, you take care of the immune system. They look after each other, and you look after the immune system, you look after everything else. So it becomes a very important system. So part of the mechanism seems to be through an activation of a cascade that stimulates macrophages systemically throughout our entire immune system, which is beneficial. So it stimulates a protective immune response throughout our bodies. And they propose possibly this pathway, but the research for this actually is still ongoing. So let's look at a little bit of work in horses where we've shown, not we, other people have shown a beneficial effect. So this was studied by No-Tan 10 years ago now. And they studied standardbred racehorses in training. And they were given about 0.5 gram of Melafeed a day for 60 days. And on days 0, that's your baseline or your control, and 30 and 60, they performed a standardized high-intensity exercise test, enough to generate muscle damage under normal situations, which is what you want because you're trying to test the system. And they measured, among other things, the plasma creatine kinase, which is a marker of muscle breakdown, erythrocyte, that's your red cell superoxide dismutase, so it's one of the superoxide dismutase isoforms in the body. And if it's upregulated, it's highly likely the other isoforms are also upregulated. And they also did a blood resistance to hemolysis test. And so this is how easily or how hard it is to break down the red blood cells. If there's membrane damage, then it's relatively easy to hemolyze them or break them down. So at the end of the 60 days, when they did this test, they found that the red blood cells were more resistant to hemolysis. So that implies that there was an increase in antioxidant systems that allowed the membranes to be stronger. So you see this in red cells, and we also see this, importantly, in muscle. Because in the animals that ate the Melafeed, that consumed the Melafeed as part of their diet, the plasma creatine kinase, this muscle enzyme that shows up in blood when the muscles leak, it remained unchanged, but it increased in the placebo group, which is what you would expect to see with very high intensity exercise. So this study showed very clearly that there's beneficial effects on mitigating the damage that's associated with very high intensity exercise. So that's the type of response that we're looking for a beneficial effect. So they concluded that the oral SOD supplementation, i.e. the Melafeed supplementation, makes the membranes less permeable during stresses associated with regular. Let's summarize. That takes us to the end. We know now that oxidative stress is not avoidable. It's an important part of our everyday lives. But the detrimental effects of oxidative stress, they can be controlled to some extent by the ingestion of nutritive antioxidants. Nutritive antioxidants that stimulate the body's natural defenses, such as the way that Melafede works, they are more effective than any of the known secondary antioxidants. And indeed, some of those secondary oxidants are effective and they're widely used, but these are actually better. Many secondary antioxidants, vitamin E and creatine, when ingested, often don't have effects that are consistent with claims. So this is because sometimes a lot of the research has been done on cell systems. It's not been done in the animal. And so it's really a little bit difficult to extrapolate, and one has to be very careful with claims. But there's so much stuff on the internet these days, so many different products, hundreds of products with these things making claims, and we have to be very careful and very judicious about how we interpret those claims, because most of them are not substantiated. We know that when we ingest the antioxidant enzymes like SOD and Melafede, that they don't get absorbed into the blood intact, and they exert their effects through different interesting mechanisms. For most nutritive antioxidants, the dose, the duration, and the outcome measures have not yet been adequately determined. There's a lot of work that needs to be done for many of them. And Lallemande has been very proactive in generating research to support some of the findings that they've seen. And we know from this very extensive research program that ingested Melafede has beneficial effects, and that these are increasingly tied into very good data respecting the dosage that the animals are taking, the duration over which they take those dosages, and the actual outcome measures. And the mechanisms are still under investigation. So thank you very much for your time, your attention. I hope you were able to get something out of this. Thank you very much.